Human macrophage biology research has been limited because it relies on collecting cells from donors. This protocol shows a method in which we can produce human macrophages from pluripotent stem cells in the laboratory.
This is a robust protocol that can be scaled up to produce macrophages in large quantities for cell therapies or research.
Pluripotent stem cells can be genetically manipulated, so in this protocol we can generate macrophages with a specific phenotype, fluorescent tags, and also macrophages to model disease states. Visual demonstration of this protocol is useful because the use of the Easy Passage tool won't be familiar to many researchers. Also, the level of care needed when replenishing media and harvesting cells is hard to put into words.
When the cultured cells have reached 80% confluency, replace the spent culture medium with 1.5 milliliters of fresh medium. Holding the culture vessel in one hand, roll a disposable cell passaging tool across the plate in one direction. All blades in the roller must be touching the plate. Maintain uniform pressure while rolling. Repeat rolling in the same direction until the whole well has been covered. Rotate the culture vessel 90 degrees and repeat rolling. With a sterile pipette, use media in the well to dislodge cut colonies. Aspirate cell start and replace media. Transfer cells at a 1:4 ratio onto pre-coated wells prepared as described in the manuscript. To begin the differentiation process on day 0, add 2.25 milliliters of stage 1 media into 2 wells of an ultra-low attachment 6-well plate.